Good. So I would like to welcome you all to this new webinar from Scilife. Okay, as I've commented, the topic of today is Annex 1. Okay, and today we have Claudia Weber. She's an experienced senior life sciences consultant at QBD. I will present her later on. But before, during this introduction, I'm going to explain you the different parts we have with this webinar. The training will be provided by Claudia. It will last about 45 minutes, right? And the topic is the new amended guideline, Annex 1. It is called Manufacture of Sterile Medicinal Products. And this guideline was amended and it will come into operation on 25th of August, 2023, except for point 8.123, which has been postponed until 25th August next year. And just to focus on the issue, Annex 1 has been amended because mostly the science, behind the sterility manufacturing is constantly evolving. It is changing, and new manufacturing technologies, new innovative technologies have emerged. So guidelines have to keep pace with the changes. Annex 1 will have a high impact on quality assurance and also with regulatory compliance in the manufacturing process of sterile medicinal products. And today, Claudia has prepared a training session that will empower you with the knowledge and with very important insights which are necessary to drive compliance in quality within your organization. All right. Claudia will delve into the background of Annex 1. She will also provide an overview of all the significant changes that has brought about with the 2022 revision. She will also explore contamination and control strategies, which are really important in this guideline. And she will examine the regulatory expectations surrounding this critical guideline. I want to explain you that at the end of the training, we will have about 10-15 minutes for the Q&A session. You will have time to ask any question you have, any question that appears to you during the training. You can use the Q&A session. You can find it below the screen. And I will collect these questions, and Claudia will make all her efforts to answer as many as we can. If any remains unanswered, don't worry, because we are going at Scilife and QBD, we are going to prepare PDF document with the most important issues about this training. And also we will add the Q&As, responses, and also additional information that you can use at your work. Okay, with that said, and with any further delay, I would like Claudia to introduce herself. Good morning, Claudia. Thank you very much for being.
Good morning, Angel. Thank you. My name's Claudia Weber. I'm a senior life sciences consultant at QBD, Quality by Design. Started my career about 25 years ago. Spent my entire career in pharma, in all three pillars of pharmaceutical production and QA and QC. So most of my focus has been on aseptic and sterile production. So hence the question from Scilife to host this webinar for you. As Angel has said, we will delve into the background. I will give you an overview of what is currently in Annex 1. We will talk about the contamination control strategy and give you some regulatory expectations. And we will end with the Q&A session. Now for the background, it has been revised. It's been revised for the 5th time, if I can remember correctly. The last was in 2008, which of course most of us know. Major changes in that version were alignment with ISO 14644, media simulation, bioburden testing, and most importantly, the capping of files. That files were not integrally closed when not capped. So that provided a lot of improvements for sterile manufacturing. Also, regulators and inspectors found over the past 10 to 15 years that better regulation is needed because companies were struggling with the interpretation of the guideline, and also shortages, recalls, and recurrent GMP non-conformances or non-compliances were troubling due to lack of process understanding, and they had seen no significant improvement over time. So that led EMA and PICS and WHO to do a complete rewrite, also incorporating advanced manufacturing technologies, also detection technologies for monitoring and detection for microbiological methods. And GMP have advanced considerably, notably ICH Q9, Q10, and ISO 14644. To give you a glimpse of the renewed focus, it has expanded from 16 to 51 pages, not counting the glossary, to give you an idea of how much more descriptive the annex is. It has multiple new and extended topics, and the focus is on risk management and contamination risk management in particular. To show that the word contamination is mentioned 35 times in the old version and 137 times in the new version. The same for risk is mentioned a lot more often, over 7 times more often. Contamination control strategy obviously was not mentioned in 2008, but is mentioned regularly in the new version, and the word monitoring also 5 times more. So that will give you a glimpse as to the renewed focus of Annex 1. So what exactly is in the new Annex? I will go over the 10 different sections of the Annex and explore some of the major issues that arise. The scope. The focus is on quality risk management throughout the entire document. So QRM does not pertain to separate sections. It applies to the entire document. Its main purpose, of course, is setting standards for sterile products and the added applicability to production of non-sterile products, which will pose or might pose a challenge to those wanting to apply parts of the annex. Section 2 is principle. It displays the ideology behind Annex 1, and essentially, of course, its sterile products should be produced in a qualified environment, trained personnel using conscious, traceable processes, and control of materials. So basically, that is the entirety of Annex 1, or rather GMP, in one sentence. But there are some core principles in Annex 1. The Annex applies to the entire supply chain, manufacturing, packaging, and logistics. Now, note that manufacturing for Annex 1 starts at selection of your suppliers. So that means that purchasing, logistics of your starter materials, QI, is also considered part of manufacturing. Second is a focus on the contamination control strategy, which we will go over in more detail later on, but is referred to throughout the entire document. Annex 1 states minimum requirements only. You have to know your process well enough to know where the risks are and maybe if you should do more than Annex 1 states. Qualification or validation is no longer a one-off event. Continuous monitoring according to Annex 15 and continuous verification to assure maintenance of the validated or qualified state. And of course, a process of continuous improvement should you venture outside of the established parameters. Personnel must be well trained. That is not a new requirement, but it is under higher focus in the new annex. Again, the mention of quality risk management and should be incorporated in the QMS. Also, this is not new for parts of Annex 1, but now it applies to the entirety of the document. It may be applicable to non-sterile products, should you choose as a non-sterile product manufacturer or an API manufacturer to choose to adopt a section, one or more sections of this annex. Be sure to document exactly what sections you are using and why, and how you are using them and how you are complying to those sections. And there's an increased focus on new technologies and systems. So that could be barrier technologies, but also robotics and rapid testing technologies. Part 3 is the pharmaceutical quality system. It will almost, of course, should be read in conjunction with GMP, not QMP, but GMP Chapter 1. Facility personnel processes monitoring should be assessed by quality risk management. Here it is again. QA oversight is explicitly addressed. Also for CDMO, for example, where QA oversight is may be somewhat murky, but as the final manufacturer or client of the CDMO, you are responsible to incorporate every bit of contamination control into or to be able to explain every part of contamination control, also the ones at your manufacturing partners. CCS, or the contamination control strategy, should be part of the management review. It should be a living document. And if you use it well, it may be quite a challenge to draft it, but once you have it, it will help you in case of deviations, in case of change control. So it's not just something that Annex 1 desires or requires, but it is actually a tool that can help you. Section 4 is premises. Of course, the clean rooms. There's a recommendation to use new technologies to prevent contamination. So that would be the RABs, the isolators, but also the robotics, the automation, to basically reduce human or manual intervention in processes as much as possible, to reduce error or to reduce contamination. Grade A is described as the critical zone for high-risk operations. Unidirectional airflow. It no longer has to be laminar, but it must be unidirectional. And limited access into grade A areas. You do not want the cleaning lady to access your grade A. And trust me, I've seen it happen. Grades B, C, and D are designated the clean rooms. They have preferably unidirectional transfer of materials and personnel into these areas via airlocks. If not possible, then there should be procedures in place to prevent contamination control, and these of course, should be part of your contamination control strategy. There's heightened attention paid to airflow pattern visualization. For grade A, of course, there is the need to establish evidence of the unidirectional airflow and absence of any turbulence that might prevent the ventilation from removing particles from your critical zone. And for the lower grade areas, you need to show that there is no ingress of contaminated air from lower grade areas. So there's also a significant expansion of the sections describing RABs and isolators. And there is some background as to what is the difference between RABs and isolators. You do not want to qualify a RABs as if it were an isolator or the other way around. This is a depiction of the classic box in a box for sterile production. Of course, we have the class A, and the background is a sterile background in case of a conventional flow hood. In case you have an isolator, then your background might be a C or a D. These clean rooms and clean air equipment must be qualified. So there's an explicit link to ISO 14644 that explains it should be classified, the particle count, filter integrity testing, airflow visualization, air pressure testing or air pressure differentials, microbial contamination, temperature, humidity, recovery and containment link testing. These are all in ISO 14644 and are explicitly mentioned in Annex 1. Now, classification should take place both at rest and in simulated operations. You do not have to do this during regular operations because these actions, classification actions, might have impact on your normal operations. So it's perfectly fine to do these during simulated operations. There's now an explicit time interval, maximum time interval for clean room requalification, 6 months for grades A and B and 12 months for grades C and D. That is not really a new requirement. It is for Annex 1, but it was also mentioned in ISO 14644 in the past that the previous annex did refer to. Now, there are some changes in the classification requirements. The 5 micrometer particle is no longer has specified limits for grades A and B at rest. If you should desire, well, of course you don't, but if you should have a need to, they should be that should be indicated in your contamination control strategy based on your historical trends. If your trends show that it is necessary for you to keep measuring them, then you should. If your history shows that they have no added value, then you should specify that in your CCS. Values for 5 micrometer for B, C, and D have slightly been changed. For B, they used to be 2900 and now they're 2930, so it's a minor change. For grade D, the in operation limits were changed from not defined to not predetermined. The difference being not defined, meaning you don't have to, never mind, to not predetermined, being you have to set the limits yourself. So there is a significant difference there. Also, microbial concentration levels must be determined during qualification. There used to be an average of -1. It did not tell you how many values you had to average that must be smaller than 1. Should it be the last 5 or the last 100 values? And basically, industry practice was it should be 0. So that's why this was incorporated in the new annex. The other values have not changed. For section 5, equipment, new is that indirect product contact parts should be sterilized. That may require extensive process changes, especially in cases of wraps and isolators, where indirect product contact parts may be affixed inside the system. But the default regulatory expectation is sterilization. There's not much guidance as to what you should do if this is not possible. So as directed, use quality risk management and set your strategy based on risk management. And if you are in doubt, contact your regulatory agent, your local regulatory representative, and ask for help. There's a new chapter on utilities that was only mentioned as a on a side note in the previous annex, but it is materials or equipment that come into direct contact or influence it directly. Water systems, water for injection, purified water, steam for sterilization. The steam quality test, or there was a quality requirement, it should be of sufficient quality. And now there is explicit requirements for steam quality testing, dryness value, compressible gases, and so forth, that have to be measured periodically. Also, compressed gas and vacuum and cooling systems, such as your HVAC, have now been described in more detail. Personnel, there's a focus on training and gowning. This is a major issue, or an issue. It's a major focus. Training and having trained and experienced people on working on every aspect of your production. So not just on your shop floor, but also in QA, also in your logistics department. So there's also training in aseptic handling is mentioned explicitly, as opposed to the previous annex. Also operate the qualification to enter grades A and B. Now there's some additional requirements or considerations. It's facility SOx. No one ever stopped to realize how many particles are generated when people change their socks. Sterile goggles, and a maximum time that cleanroom garments can be worn must be defined. So you, as a manufacturer, have to define how long garments can be worn. Also, there is a mention of glove disinfection. They should be regularly disinfected, Annex 1 says. It does not tell you what regularly is. It does not tell you how to disinfect your gloves. There is, of course, it is common practice to qualify and validate disinfection methods for surfaces. I would advise you to do the same for gloves. How much disinfectant is to be used, which disinfectant is to be used, how are they supposed to apply it, and so on. It's a crucial step, so include it in your contamination control strategy as well. as it is to be done prior to interventions. So it's a critical step, really. The old annex said, well, operations and grades to be performed in grade A, it's rather minor, and it has been extended in the current annex. Also grade B was added, just to give you an idea of how much more prescriptive the new annex really is. Section 8 relates to production and specific technologies, focuses on the design process. And again, there's a preference for technical solutions, robotics, and RABs and isolators. So removal of people from the direct vicinity of the process as much as possible. Also, it describes interventions, that new interventions, of course, should be assessed prior to being allowed. And all interventions should be covered by your aseptic process simulations. Sterilization techniques have been described in more detail with an increased focus, not just for Annex 1, but also for FDA, for load configuration and validation. How do you configure your loads? Most companies will use minimum and maximum load based on worst case or a matrix matrix program. So you must clearly define how you determine your worst case loads. Factor usually used is mass for the load, but factors that can also be of importance are geometry. If you have a long, thin bore tubing, it can be very hard to remove air and be very hard to validate. And also draining capacity. Again, sterilization of direct and indirect product parts is, or product contact parts is mentioned. There's an expansion of the blow fill seal and form fill seal sections. I will not go into that. PubSit was introduced and 100% integrity testing. Now, PubSit was and still is a matter of extensive debate. Now, why would they want you to perform PubSit? To detect flaws caused by production and filter preparation that are not caught by post-use integrity testing. And also a phenomenon called filter flaw masking, that there is a minor flaw that is masked by a product component and can also cause a false pass in the post-use integrity testing. There's risks to PubSit because they are complex and they require actions and added components to the testing setup that leave room to introduce errors that would otherwise not exist, like operator errors, leaks, non-integral added components. There's, if you see in the next slide, there's two filters in the center and there's two testers for two filters in serial filters, but there's a lot of added components. There's a lot of added sterile connections because you have to prevent microbial or the bioburden overload on the upstream of the filter, and you have to prevent sterile or contamination of the downstream of the filters, downstream side. So it's a lot more complex. So why do we want to do it? Or why do the people from EMA want you to do it. The current controls during manufacturing, transport, and installation are not sufficient to protect against minor flaws. So if there are flaws in your filter and you cannot detect them, then you might have a production issue. So we need unbiased risk assessment if it is not possible. If you have a very small volume of product to be filtered and you need to wet the filters in order to perform PubSit, then you might run the risk of diluting your product. So that would be a reason why you should, why you could choose against performing PubSit. And also the problem is we don't really know how often filter flaw masking occurs. So filter production or filter manufacturers are working around the clock to gather data regarding this issue. There's a subsection added regarding single-use systems. Usually it's outsourced and you buy them pre-assembled and pre-validate or pre-sterilized. They will lead to additional validation parameters for your process, like leachables, extractables, reactivity with your product. Requirements for supplier management, because you need to verify that your supplier has sufficiently validated their production process, their sterilization process, etc. Then in case now with the heightened focus on environmental control, you should take into account if materials are biodegradable or recyclable. If they are recyclable, how are you to incorporate this recycling in your normal waste processing? There's a subsection of closed systems that was added. Reduces or eliminates the need for aseptic connections, single-use, pre-sterilized, ready-to-use systems, or you can prepare them in-house and sterilize them yourself, which of course you have to validate and perform integrity testing and also interaction with your product. But it is highly recommended, by the way. So it may come as a challenge, but they are moving more and more towards closed systems and single-use systems. Section 9 is variable and environmental monitoring. A monitoring program should be part of your contamination control strategy, which is not very surprising. There is an explicit description of what should be in your monitoring program. Total particles, viable particles, things like temperature, relative humidity, other specific characteristics. Pressure differential could be one of them, and also your aseptic process simulation, should you do aseptic production, of course. Alert and action levels are based on results of PQ testing in case of new facilities, or trend data when you have them, when you have sufficient data. Alert and action levels should be subject to periodic review. It's not a one-off action to determine your alert and action levels. You should revisit them every once in a while and see if they are still accurate. Increased focus on trending, monitoring, and also increased focus on continuous improvement. How are you trending? Increasing numbers of alert or action or alert limit excursions, consecutive excursions, things that may have a common cause, changes in flora, like type and numbers, molds, for example. And you are wise to describe in your monitoring SOP what is to be done when you have excursions. So you don't have to come up with a solution once you already have a deviation. The permitted particle counts for grades A and B for 5 micrometer samples have also slightly changed. Here they do have limits for grades A and B. And again, for also for grade A, it says continuous monitoring throughout the entire process. And it is also recommended for class B, which does not mean it is mandated, but it is expected of you to justify not doing so. And again, for class D, the values in operation are not predetermined, meaning that you have to determine them yourself. For micro, grade A, again, the same as for clean room qualification, the average is no longer accepted. And so for grade A, no growth is accepted. Personnel monitoring was added on exit and after critical interventions. Now that might be a challenge as well, because if you monitor gowning, then you would have to regown afterwards before you can continue processing. Microorganisms should be identified to species level. This is not new for most companies. It will help you determine the origin of your contamination. And the impact should be evaluated for each batch implicated. This leads probably to a list of objectionable microorganisms. Microorganisms that you do not want to see or find in your product or in your facilities. Of course, molds, but there might also be a few pathogens that you do not want to be part of your product. Also, there is what I find interesting is that there is still no limits for swaps. Swaps are to be used in monitoring of irregular services, but there is no guidance to the use of swaps in this case. Now, for aseptic process simulation is also described in a lot more detail than the previous revision. Interventions should be documented, of course, but they should also be covered in your aseptic process simulation. Your initial validation should be at least 3 consecutive aseptic process simulation covering all working shifts. That's a new one. After any significant modification that has the potential to impact sterility assurance of the product. It should be repeated twice a year, normally for each aseptic process, each filling line, and each shift. This is also much more explicit. It was implied in the previous version, but it was not this explicit. Each operator should participate in at least one successful aseptic process simulation annually. I think most of us have already done that. A new one is the consideration that should be given to aseptic process simulation after the last batch, prior to shutdown or before long periods of inactivity, to confirm that any batches produced prior to shutdown are within your valid aseptic process simulation. But it is to be considered, meaning it is not mandated, but you should justify not doing so. To the requirements, there is no growth allowed anymore. Too bad. The 2008 version allowed for 1 unit to contain growth, provided it was investigated in a certain number of units in a batch. I hope, sincerely hope, that no business has allowed any units containing growth. It describes when a failure is due to operator activity, the operator should be retrained, requalified before he regains access to the rooms and regains access to the process. And there's a very explicit explanation on what an APS plan should look like. It's based on risk assessment. Identify your worst case conditions like file size, holding times, line speed, etc. Nutrient media selection was supposed, in the old annex, it said it should basically be a good media, an aselective media to help you grow any microorganisms in your process. A new one is that it should be able to simulate your process. I don't know if any of you have tried to filter the TSB medium that the European Pharmacopoeia prescribes, but that's no joke. So it's very hard to filter. So that is why they have added that you should choose a medium that allows you to properly simulate your process. Process simulation duration, it should include startup and stop interventions, any corrective interventions, any normal interventions like replenishing stoppers and such, or bulk product, any normal interruptions such as lunch break should be incorporated in your media fill. And of course, there should be normal environmental monitoring. Section 10 is quality control, and that also focuses on personnel training and experience. That is the main focus, aside from the other subjects in there, like types of microbiological testing, sampling, release, and data review. Now, for the contamination control strategy, the goal is to exhibit, or for Annex 1, to force you to gather detailed process knowledge, know what you're doing and why. Identification of all associated potential contamination sources, meaning risk assessment, and include the entire supply chain. So start at selection of your starter material or packaging material suppliers. Assurance of appropriate controls to manage risks, also your risk management, and refer to all measures that you have taken. You have most likely an SOP describing the way you handle sterilization, the way you handle monitoring, the way you handle sampling. Those are documents that you need to refer to. Types of contamination to consider, of course, sterility, bioburden, endotoxin, particulate matter, but maybe also, if applicable, viruses or cross-contamination. All these separate parts most companies already have in one way or another. The new part of the contamination control strategy is that you have to describe how these various control measures interrelate. How does your cleaning influence your material control? Or how does your design, your facility layout impact your monitoring? All these all these aspects, they are interrelated more than people realize. And this is where you should consciously determine how they are interrelated. And again, it should be part of management review. And it is a living document. No one is expecting you to draft the perfect contamination control strategy at the first go. Just start and improve as you go along, as you would with any other living document. So again, it's not an entirely new concept, but describing the interactions is in fact new. This list is non-exhaustive. This is the list that comes literally from Annex 1. It's supposed to give you an idea of the elements that should be described in your contamination control strategy, if they are relevant, of course, for you. If you have no outsourced surfaces, then of course there is no need to describe them, but you would have to describe that you don't have them. Others will most likely be relevant. And also you should describe how you strive for continuous improvement based on the information from all these systems, based from monitoring all these systems. And again, this is not exhaustive. So maybe if in your case, virus safety or adventitious agents or pest control are relevant, then you should describe those here. So how to start developing, you have to understand the impact of a change in the elements of the contamination control strategy. Identify the risks for product or patient safety. That is your risk assessment. That is the basis of your contamination control strategy. You have to thoroughly describe your process and assess the risks. Now, of course, when you have identified the risks, you have also developed measures to eliminate or reduce them to an acceptable level, which usually means that you have procedures in place for disinfection, cleaning, sterilization, and these are the measures that are described to eliminate the risks. You have to perform and implement these measures. It's one thing to write an SOP, but then you have to make sure that it is actually performed. Ensure the resulting tasks are actually implemented. And be sure to do that under change control if necessary. Document the evidence, of course, and evaluate the effectiveness. That again is the continuous review, the continuous verification and identification of possible improvements. That it keeps it a living document. So you have to keep it alive. So this is a schematic that is from the ECA, the European Compliance Agency document, how to develop and document a contamination control strategy. It's a very insightful document that can help you if you have to start your contamination control strategy. First is familiarize yourself with Annex 1. Know what it is that it is expected of you. Find the gaps and then remediate the gaps. Compile the documentation and of course evaluate. Now for regulatory expectations, what do they expect? In short, quality risk management. Anything you do, anything you write, anything you say must be based on risk assessment. does not have to be an FMEA, can be other methodologies or very informal methodologies. Just document why you do what you do. In-depth knowledge of one's processes, the entire supply chain, again. Identification of risks and remediation actions, there it is. Advancement of new sterile manufacturing technologies, if possible. Of course, if you have a facility that is working perfectly fine, you are not going to remodel just to introduce robotics. But if you're moving to new facilities, remodeling, replacing equipment, then you should consider using new technologies. And you are expected to justify not using them. Of course, there's the increased focus on monitoring and periodic review, not just environmental monitoring, but also equipment monitoring, process monitoring, periodic review, and continuous improvement. And of course, you have to have the contamination control strategy. So to implement, you create a gap assessment and create a realistic remediation plan to address any non-compliances in a timely manner. So that is it for me. Now it is up to you.
Okay, Claudia.
Back to you, Angel.
Yeah, right. Thank you very much, Claudia. Yeah, I think your expertise and insights on this topic, I think they have been really invaluable today. Thank you very much. I think you have provided us with a deeper understanding of this crucial Annex 1 guideline and the implications on sterile. Thank you. All right, so this concludes the training. So we have now some spare time, some, well, 6 minutes for the Q&A session, right? So we'd like to open up for questions. You can use the Q&A button, which is under the screen in the Zoom bar. So let's go for the first one. Yeah, Claudia, what about if we cannot make the deadline for the Annex 1? What can we do?
Of course, you have to, or I would hope that anyone already has, since we're 1 month from coming into operation, that companies have completed their gap assessment and then they have assessed criticality and impact of resolving these gaps. If a gap should, resolving a gap should just take a documentation change, or should take an entire project, for instance, that includes change control and such, create an implementation plan, create CAPA, and document it. If you have a valid reason for not complying or not being able to make the deadline, like your equipment is not here or your entire QA department has left the building for the past year, you should have that documented. Contact your regulatory representative if necessary, and be proactive in letting them know that you may not make the deadline. It will probably help you gain more leniency from them should you be audited.
Okay, good. Right, thank you. All right, so let's go for the second one. Annex 1 does not specifically refer to the cell and gene therapies or the ATMP products, right? So what will be the MEA approach during inspections in relation to Annex 1 with for such facilities?
ATMPs are covered in section 4 of GMP. That is a standalone section of documents. But if ATMP manufacturers follow PICS Annex 2 or Annex 2A, that refers to PICS Annex 1, which is identical now to EU Annex 1. So in that respect, you will have to, if you follow PICS, then you are expected to follow Annex 1 or PICS Annex 1. So that in that case, you will be expected to follow Annex 1.
Okay, good. Another one is Annex 1 refers to the use of new technologies or containment systems such as isolators or wraps. So can you explain us shortly which is the difference between isolators and wraps?
Well, there's been a bit of debate about what is a wraps and what is an isolator, most likely because a lot of wraps systems are starting to look more like isolators. So they have they were designed in origin as an advancement to a old-fashioned run-of-the-mill laminar flow hood. So it was a laminar flow hood with doors, for instance. Now they have gloves, but they are regarded as an advancement to a laminar flow hood, basically. And that is why they still are to be placed in a Class B background, even though they might have gloves. Now, the confusion when they have gloves is that some may start qualifying RABS gloves as if they were isolator gloves. But isolators are supposed to establish a complete segregation of the Class A environment from the background environment, which requires a lot different things from the gloves or the separative system than it would from RABS. So it would not be advantageous to qualify RABS gloves as if they were isolator gloves. So in short, the isolator is supposed to provide complete segregation where the RABS does not need to. If that answers your question.
Okay, we have just 1 minute, so let's go for another one and let's make the wrap up. We have a question from Armin. Is a PubSit required also for manufacturing of investigational medicinal products?
If you should, investigational products, I think has a separate annex. I am not quite sure. I would have to dive into that. So to give a proper answer, if you choose to follow Annex 1, then yes, you should perform PubSit. If unless it's not possible, like we discussed, then you have to verify why it's not possible. But I would have to go into that. I will get back to that.
All right, so no problem, because we're going to collect all remaining questions. So yeah, Claudia will be able to answer all of them. So, and we will send you a PDF document with the responses of those questions and any additional information. All right, so we have to wrap up because it is a 1-hour training. So I want to appreciate Claudia. Thank you very much. And also everybody for being here and posting a lot of questions we have today. You can visit our websites at QBD and at Scilife and let us know if we can help you in anything. Okay, so you can get in contact with us at these links. Okay, so yeah, we appreciate you for being here. Thank you very much, and I hope to see you in the next training sessions we perform every month. Thank you very much, and see you another day. Have a nice day.
Have a nice day.